of the biochemical reactions resulting in mousy off flavors, in particular the production of ACTPY, which is acetyl tetrahydropyridine, and ACPY, which is acetylpyrroline. Both of these molecules result from the deamination and decarboxylation of an amino acid. In this particular case, lysine results in the formation of ACTPY, and ornithine results in the formation of ACPY. During this process, you make this heterocycle intermediate that can be acylated using an intermediate from the heterolactic pathway, for example. And so here's the acyl side chain here that's added. And this is the mousy, stinky product from that process of deamination and decarboxylation of lysine. Pediococcus species can produce biogenic amines, which I've talked about, but they also can produce something called ropiness, which I've also talked about. And the ropiness is basically a texture defect, primarily caused by the enzyme glycosyltransferase, which the bacteria produce. And so the ability of that enzyme to polymerize glucose monomers into chains causes that thickness and viscosity. What's cool is that the microbes actually have this enzyme on a mobilizable plasmid, and so they can share this capability with each other. It's the same kind of thing with a microbe when it has an antibiotic resistance plasmid, and it can share that plasmid to make other members of its species resistant to that antibiotic as well. So Pediococcus, as you can see here in this electron micrograph, as the name suggests, are small little gram-positive cocci. Another spoilage microbe is Xenococcus. But of course, this is a commonly used microbe to inoculate for malolactic conversion, so it's also a good guy. It's just that some native strains have the ability to produce biogenic amines, which is why I said previously that we really need to characterize your malolactic strain to make sure it won't make these foul-smelling and allergy-inducing, etc., biogenic amines. Enococcus can also produce high levels of VA after a malolactic, particularly if you have a stuck or sluggish primary. And so again, if Saccharomyces is gobbling up all the sugar and leaving some residual, then Enococcus can cause spoilage. Acetic acid bacteria are unique in the sense that they are gram-negative rods. All the other organisms that I've talked about or at least all the other bacteria that I've talked about are gram-positives. So these are aerobic organisms as well, and so they can grow when there's a bit of headspace left in a barrel, for example, during aging. And so the three main genera are Gluconobacter, Acetobacter, and Gluconoacetobacter. These guys obviously produce elevated VA in wine or juice that has a little bit of oxygen available for them. They can also produce ethyl acetate and aldehyde. So the good thing about these things being gram-negative is that if you suspect elevated VA or you've measured elevated VA, you can dip in and do a quick gram stain, and if you see small gram-negative rods, that's pretty diagnostic. So here's a little bit about their metabolism. Again, the organisms are oxidizing ethanol to acetaldehyde and then acetate. And so here's the ethanol that your primary yeast produces, and the ethanol gets transported into the cell, and they have an enzyme called alcohol dehydrogenase that allows them to oxidize the ethanol to acetaldehyde, and ultimately an aldehyde dehydrogenase that allows them to further oxidize the acetaldehyde to acetate. When the oxygen conditions are really low, the microbe will spit some of that into the medium. And this is typically what happens in wine fermentations where acetic acid bacteria are present, because it's mostly a low oxygen environment. So they'll spit some of this waste product into the environment. Some of that material, some of the acetate, will ultimately be used to make acetyl-CoA, which is an essential molecule that gets fed into the TCA cycle where the bacteria make energy. Some of this ethanol and acetyl-CoA can also combine to make the nail polish remover, basically, which is then spit into the environment as well. Another spoilage microbe is Brettanomyces, the yeast Brettanomyces. Now, in beer production, these guys are are desirable. You know, libertine type places that like sours and so forth rely heavily on Brettanomyces, but in wine, these guys produce defects. And here's just a few of those. And then the names of the compounds are here. This next slide, oh, actually, I should just mention that, you know, morphologically, these guys are kind of what we call boat-shaped, so they're pretty easy to pick up through a simple stain, just looking under the microscope if you start to smell the bready off character. So this slide just shows you a little bit of the biochemistry of Brett reactions. And so in this particular case, you're looking at a hydroxycinnamonic acid derivative, so this backbone here, which is universal, and then this R group circled here can either be an H, an OCH, or an OH. And you know, if it's an H, for example, then that's paracoumaric acid. Again, these molecules are derived from the grapes, and so if there's an abundance of them and Brett happens to be present, it can convert these into foul-smelling compounds. So if the R group is an H, then we have paracoumaric acid. The microbe will produce phenolic acid decarboxylase, which pulls a carbon dioxide group off of the paracoumaric acid to produce vinyl phenol. The microbes then produce a vinyl phenol reductase, and in this particular case can ultimately reduce the molecule, the vinyl phenol, to 4-ethyl phenol, and then the 4-ethyl phenol can be belched out into the medium. And so the same can be said for these other molecules as well. And so we know that 4-EP, for example, has a medicinal kind of Band-Aid smell, and 4-EG, for example, has a smoky kind of clove type aroma. And 4-EC, I've heard this, I've never smelled it personally, but I do work with Brett, but I've never really smelled it as a kind of a horse blanket or a barn type smell. People say I can't pick it up because the problem is when you grow Brett, it usually produces 4-EP, and that's usually the dominant smell, so you mostly only smell the medicinal Band-Aid smell. But again, the sensory level for these things is really low. Like for 4-EG, it's about 300 micrograms per liter before you can start smelling the smoke and cloves. And 4-EP is about, I don't know, maybe 600 micrograms per liter before you can start smelling the medicinal Band-Aid smell. And so again, and it also depends on the wine and so forth, because these thresholds can be lower or higher depending on the wine. Another microbe, Zygosaccharomyces, morphologically looks very similar to Saccharomyces, these little football-shaped things that you see here, and you can see one of these guys budding. But they can cause increased turbidity and CO2 in bottled sweet wines, for example. They can form late blooms and so forth. One of the problems with them is that they're tolerant of SO2 and they're tolerant of high ethanol. And so that's one of the reasons, and also low temperatures. And that's one of the reasons why you can see some of this late turbidity and CO2 formation from these types of microbes. And they can also produce acetic acid and acetaldehyde. Saccharomyces is obviously our primary yeast, but if allowed, if it's not filtered out, for example, then the microbe can cause turbidity or CO2 in bottled wines if there's any residual sugars left behind. This is an overview of the biochemical pathways that might lead to sulfate reduction and the formation of sulfide. So this is the normal pathway. This is what we would want to happen, where there's a sufficient amino acids and ammonium that forms the nitrogen pool in the cytoplasm of a yeast cell. And then the yeast can take this nitrogen and, through a series of steps, produce molecules like O-acetylserine and O-acetylhomoserine, combined with sulfate from the environment that is ultimately reduced to sulfide, and these guys can make sulfur-containing amino acids like cysteine and methionine. So that's the ideal situation. But in this particular diagram, what you're seeing is the microbe the yeast being starved for amino acids or ammonium ions, and so the nitrogen pool is depleted, and this process then of making O-acetylserine and O-acetylhomoserine are blocked. So as sulfate comes in and is reduced to sulfide, this sulfide is toxic, and so the cells have to get rid of it. And so they'll belch it back out into the medium, and there's where you get a lot of the rotten egg or foul-smelling side aromas from sulfur and amino acid metabolism gone wrong. Usually we can see this kind of as a in a prolonged exposure to leaves, for example. If the microbes are left on the leaves for a long time and they've depleted the amino acids, eventually they'll start pumping out sulfide. I've seen instances where excess elemental spray in a vineyard, used typically for mold control, ultimately had an effect on sulfide production later in the fermentation, because there's the extra sulfide that the yeasts were exposed to. And so usually what's done is copper sulfate fining, for example, to pull out some of the excess sulfide. The copper sulfate will dissociate at wine pH, and so the copper ions can actually bind to the sulfide and precipitate that out of solution as a sort of a black precipitate at the bottom of a tank.